Thank you, Dr. Martinez, and thank you, Dr. Silva. It's really a pleasure to be here again. It's always nice to be in Puerto Rico, and especially when the weather is good. And I'm very pleased that so many of you have taken the time to come and hear about this topic, which is not only a requirement both of the school and the funding institutions, but really is an important topic. It's something that I've been teaching for quite a while now. Originally got started when the NIH first made their requirements, and basically someone from the school came to me and said, "You're teaching research ethics." I said, "Okay," and that didn't sound all that interesting to me at the time. And as I started reading about it, I realized that there was a lot about this material, even as a professor, which I didn't know myself, because back when I was in graduate school, we never had courses like this. We just, we were interested in science. We're basically honest. We went to the laboratory of a professor who was also interested in science and honest, and the professor would teach the students how to do things, and we would do them, and we would develop ourselves as scientists. And somehow along the way, we were supposed to learn how to be responsible scientists. But that was all kind of just happening by the wayside. There really wasn't any instruction. And it was very easy for something not to be covered and for mistakes to be made. I don't think that the reason for this is that we're going to take evil people and make them good. I think basically all of you are interested in science, and you're all basically honest. And I'm sure that if there were a dishonest person here, that my lecture will not make good people out of them. But on the other hand, even for good, honest scientists, there are a lot of decisions that have to be made about how to do things. And these decisions need to be thought about carefully, and you have to really consider what is the right way, what's the best way, what's going to have the best possible outcome. And you have to think about those kinds of decisions just as seriously as you think about the strictly scientific problems of doing your assays or cloning your genes or whatever it is you're doing for your science. So today I'm going to be covering 4 topics: data acquisition, peer review, conflict of interest, and collaborative research. And I want to encourage you to think about how to make the right ethical decisions. Now, one of the reasons why in courses like this there usually is not an examination is that for many of these questions that will come up today, there really isn't a right or a wrong answer that's absolutely black and white, but rather there are different ways that people could approach something. Some will have better outcomes and some will have worse outcomes, and it's important to think about those. Clearly, there are some things that are absolutely wrong and some things that are absolutely right, but there's a lot of gray area in between, and very often what we have to think about is, under the circumstances, what's the best possible course in this circumstance. Each circumstance can be a little different. And so I want to encourage you to think about these things. So as I'm giving this lecture, I'm going to try to illustrate it with a number of cases that we'll discuss. That means you folks too, not just me. And if at any point something comes up that you'd like to discuss, please raise your hand, interrupt me, I don't mind. And because the most important things to talk about are the things that you want to talk about. So don't feel intimidated, and maybe by later in the day some of you will even fill in the front half of the lecture hall, although I'm used to speaking to people in the back. It's all right. So this is material that you've seen before, I'm sure. That the basic definition of scientific misconduct is fabrication, falsification, and plagiarism. Other definitions have been used, but those definitions are really no longer really much in fashion, because the real problem was a lot of things would happen and people would say, well, this isn't fabrication, falsification, or plagiarism, but it sounds wrong to me. And it turns out those kinds of issues have never actually been successfully prosecuted. So many of the regulatory groups are starting to think, well, maybe we really can't define that, because it turns out that in different branches of science and different institutions, just how science gets done in different communities is different. So it's very hard to say things other than those the top three there are a problem. And the important thing is that this relates to the proposing, performing, or reviewing of research or reporting research results. So we're only talking here about research, which means a systematic investigation, including development, testing, and evaluation designed to develop or contribute to generalizable knowledge. Generalizable knowledge usually translates as publication. So I imagine here probably everyone here is a graduate student, but often I find some medical folks mixed in. And this does not relate to clinical practice. If a doctor tries an unproven treatment, that's not research, not unless it's part of a scientific study. If someone is doing a program or course evaluation, they may be using what sounds like scientific techniques, but that is not research unless there's a plan to publish the results. And also teaching a course is not research. So everything we're going to be talking about today only relates to research. And of course, the reason why we're teaching responsible conduct of research is because science is really based on trust. We trust each other's peer-reviewed data. When we read something in a journal, we may disagree with it. We may think the interpretation is all wrong. We may even think the experiment was done incorrectly. But we basically have the assumption that whoever wrote this article really did what they said they did, and these are the results. And then we can interpret those results however we want. All of science would break down if we picked up articles and we didn't believe what was written there. So that's very important. And of course, we are all honest, but there have been scandals, and the real problem with those scandals is, number one, of course, bad information will get published. That can be really a problem for scientists. If you're trying to reproduce an experiment and you can't because that experiment was somehow fraudulent, you've got a real problem there. Not only, it's more of a problem than just that the article wasn't good. And of course, being in the biomedical sciences as we are, if something is done incorrectly, that can have an impact on patient care. That can be a matter of life and death. So that really is important. So it's really important that we prevent scandals and that also keep public faith, because what happens is we all directly or indirectly depend on the public to support us. We are working here as we are in a public university. Whether we're in a public or a private university, our research is funded by federal funding agencies or charitable agencies. And those agencies, either directly or indirectly, get their money from the public. And if the public sees article after article in their newspaper saying scientists are dishonest, they say, well, I don't want my tax dollars or I don't want my contributions going to support those crooks. And then we're all in hot water. So it's very important that we maintain the public faith in the scientific enterprise. Now, I'm going to just quickly go through these. I believe that this material is on your handouts, but there are a lot of references here. If you have questions and want to go further into it, there are a number of excellent books that are available. If you have specific interests, you can look into those. There are also some very good websites. One thing that's very interesting is this Office of Research Integrity website. You can go there, and it not only tells you all the latest rules and regulations, but it will also have news about who's been convicted lately, what happened to them as a consequence of their conviction, and you can really get the latest dirt on those people who haven't been doing what we've been discussing correctly. And also, you might want to look at this update on requirements for instruction of responsible conduct of research. Until that came out about a year ago, it used to be thought that, well, if we take every graduate student in the country and we have them walk into a room like this and hear a lecture like this, that's the end of everyone's obligation, but it turns out it isn't. And one of the things that this says is that at every stage of our professional careers, there should be continued education in responsible conduct of research, but that training should be stage appropriate. So you'll probably be doing activities like this again when you're at later stages of your career, but probably not by sitting in a lecture hall. Now, another resource that you might be interested in, I'm going to today be discussing some cases, and I believe all the cases that I have today come from this handbook, which is a little out of date. It's from 1994, but basically I think that there's nothing scientifically that's terribly out of date. They wrote it kind of generalizably enough that it fits with today's science. And this book doesn't really have answers in it, but it has cases and questions to allow thought. So, and it's very inexpensive. I think they actually recently raised the price. It's now up to $15. But it's, if any of you should in the future find yourself needing a bunch of cases for courses that you have to teach in your future careers, this is a great source of that material. Of course, the school here has their program and has a lot of links to other resources that you may want to use. And I've included a few here. Indiana University and Columbia have very good programs that I've linked here. And of course, there are also good programs at a number of other places. Duke University, for example, has a very good program as well. You might be interested in that. And of course, any of you who get interested in plagiarism, this is an interesting site where you can check your own papers or other people's papers to see if inadvertently plagiarism might have occurred. Not a bad thing to do when you're submitting an article for publication. And of course, for human subjects, there are also a variety of resources that you can use. So today we're going to be talking about data acquisition to start with. And data, of course, are the basis of science. We do experiments and we get results, and everything we do depends on those results. Now, when you do an experiment and you generate results, those results are property. You have a notebook that's a physical thing, or a computer disk, or some object that has your data stored on it, no longer always paper, and that property may even be worth something. For example, it might be patentable information. That could be worth money to someone, or it may be worth something to you more valuable than money. For example, getting your degree, getting a job. So, so here is this valuable data. Who owns that data? Let's see. How many people think you own the data? Okay. How many people think your professor owns the data? How many people think the NIH that funds your lab owns the data? How many people think UPR owns the data? Well, you're right. UPR owns the data. Technically, even as a full professor at my institution, I don't own the data in my lab. My university owns that data. Just like when a grant is awarded, I like to say, like you said, the NIH has given me a grant. The NIH has never given me a grant. The NIH has given my university a grant, and my university is wise enough to think that if I wrote the grant, they'll probably let me run it. So, so it's very important to realize that the data is owned by the institution that hires you. Someone's hired you to do research, and they own the data. Now, at a university, that doesn't sound like such a big deal. But if you worked, for example, for a chemical company or a pharmaceutical company, and you tried walking out of the building carrying your notebook, there would be a security guard in the lobby. I've gone to visit people at drug companies, and if I walk out of that building carrying a notebook, the security guard stops me and says, "What's in that notebook? I want to see that, because I don't want you carrying any of our data out unless the company has given you permission to take it out." So, in a situation where it's a commercial operation, that question of data ownership can be very, very important. Now, how you store your data is also very important. Technically, and if any of you work on patentable research, whether in a university or in a company, and certainly if you work in a company on any research, you will have to not only write your data down in some kind of a format that is not erasable, and either a protected software or a hardbound notebook where you write in ink, and all your data has to be signed, written in ink, dated, and signed. Now, that can really be very important. One obvious reason it's important is for patents. Because if you invent something, you may not actually apply for your patent until some period of time after you've made the discovery. And then the question will legally come up, when did you make this discovery, and are you really the first to discover this thing, or did someone else discover it before you, and maybe you're not the inventor? Well, you've got to be able to prove when you invented it. And if you don't have a dated, signed, and witnessed document saying that this happened on such and such a date, and you didn't just say it happened on that date, but someone else signed it to say that they witnessed it, well, you have no proof, and you probably will not be able to get a patent. Now, I have a few patents. I'm waiting to make my first dollar off those patents, but I suppose it could happen someday. Other people may have been more fortunate than I have on that. But the other reason why it's very important to keep that data in a good format like this is sometimes questions come up about whether something is really done correctly or not. No less a person than David Baltimore had an associate, Dr. Imanishi, in his lab, who was a very good scientist, apparently. And at one point, the postdoc in the lab said, I don't believe Dr. Imanishi really did what she said she did in her publications. She was already a faculty member at that point. And she said, I think this is fraudulent. And there was a big investigation. And of course, they went back and looked at Dr. Imanishi's notebooks. And as near as anyone can tell, Dr. Imanishi was a very good scientist, but she was a very sloppy record keeper. And in fact, when they went back and looked at her notebooks, they discovered that some of the pages on which the crucial experiments were done had ink on them, which hadn't been invented yet on the date that was the date of those pages in the notebook. So obviously, Dr. Imanishi had gone back and made corrections on those pages. Well, that's not necessarily a bad thing, in that, supposing you discover something and then later you discover something else, you want to correct that. Well, you should, at that point, if you make any change, you should indicate what's been changed, and you should date the material that you add. So, and the best way to do that is simply to do it on another page of the notebook later on and index that. So it's very important not to have any question about when things were done. So you need accurate records, and you need to know dates for priority. Now, most of us probably don't have witnessed notebooks, but we certainly should keep them accurately dated and signed and indexed. Very important, so you can go back and find things. I know when I first started in the lab, I was very careless about that sort of thing. I said, oh, these experiments are so important to me that I will remember them forever as a brand new graduate student. And after a few months, I realized, I don't remember anything I did 3 months ago. So very important to keep good records. If nothing else, so you can repeat your own experiments, or if something doesn't work, you want to have that in your notebook too, because sometimes the experiment that doesn't work is the most important thing to keep track of, because you don't want to keep repeating things that don't work. You want to be able to go over that and say, look, this experiment didn't work, I've got it recorded in my notebook, what exactly did I do, and now I can go back and change some variable and see if I can get it to work. So you can't just throw out a page and say, this didn't work, I'm throwing out the page. No, you keep it in the notebook. Yeah, and that's why the notebook pages have to be numbered and be non-removable. You can't do lab notebooks in loose leafs, because there'll be no record if you've removed the page. If something didn't work, you can write down, this experiment didn't work, but it has to be there in the notebook. Now, since I was a graduate student, of course, one thing that's very, very much changed is nowadays many of you, especially those who are using sophisticated physical techniques in your research, may have experiments where there is no paper generated, where everything is some kind of a computerized information. And so you have to record in your notebook what experiment was done, where the data are, what kind of machine it's on, what kind of software it's on. Because again, you want to always be able to go back to that. And very often, 5 years later, someone may want to say, well, I've got a question about this particular piece of data. I want to go back and look at that, but now that computer isn't there, and people are using a different software, and you have to have some way of taking the information off that disk and making it accessible. So if you have all that information, you can go back and do that. Now, another problem that students often get into, especially when it comes time to do a talk or write an abstract or write a paper, is can you really present all your data? Now, I said this data is very, very important. But for example, when I went to write my first paper for publication, I had 2 notebooks full of data, each of which was, about 2 inches thick. And I had all that material, and I discovered that the journal I was writing my paper for had a 5-page limit. So I obviously could not put, notebooks this thick into 5 pages. So I had to decide which data I was going to present and which data I wasn't going to present. And obviously, 98% of my data was not going to be presented in those papers. So how do we decide what not to present? And that's an important question. You can't simply say, well, I did the experiment 5 times, 4 times I got a result I didn't like, but 1 time I got just what I wanted. So I'm going to write that one up, and I'm going to forget about the other ones. Because the whole point of doing experiments is you're testing a hypothesis. And very often, getting an answer that's inconsistent with the hypothesis is very important. And some of the best results I've ever gotten in the lab have been experiments that didn't work, but gave me something new to think about that was totally inconsistent with my original hypothesis. So, you can't ignore those. But on the other hand, there are sometimes things that you really can't present. If you do an experiment on some cultured cells and the cells die, well, because there was a power failure overnight, well, you obviously can't present the results with those cells if the cells are dead. But how do you decide? And the answer is there has to be a reason for either excluding the data, and if you have outliers, if you have points that look wrong, you can't simply say, I don't like how they look. The ideal thing is probably to either if there's something grossly wrong, and it's grossly wrong because there is independent evidence that just the experiment was not done correctly, you can say, well, that experiment really wasn't done, so I'm not presenting the experiment. On the other hand, if it gave you an answer that that is inconsistent with many other experiments, probably the best thing is to present it, but to indicate why you believe that result is different than all the other results. So that's very important, how you handle outliers, and that's something that the members of your research group should be talking about when you present data. So let's go over a case now, and now you'll start working on this. Jocelyn Fox, a second-year graduate student, has just begun the experimental phase of her training program. She has initiated studies of novel agents on nitric oxide synthase, most recently in a controlled experiment involving a compound identified as SR582. In her first attempt at the experiment, Jocelyn made an error in dilution, causing her inadvertently to test the compound at a concentration much higher than called for in the protocol. This yielded results quite different from those expected. She showed her data to her mentor, Professor Collins, who suggested in essence that she ignore the experiment because of the error. In testing each compound, we always stop at 1 nanomolar concentration, he said, and have been able to compare stimulators and inhibitors quite well. Jocelyn repeated the experiment, starting with the correct dilution, but then extended the experimental range all the way to 5 nanomolar concentration. And here you can see the results of the standard experiment and Jocelyn's experiment superimposed. Her plotted results were almost identical to the earlier curve obtained as a consequence of her dilution error, and seemed to confirm the inhibitory effect she observed at greater than 1 nanomolar concentrations of SR582. Professor Collins was unimpressed when shown the data and suggested that she proceed to the next phase of the experimental plan now that she had mastered the technique. So, have you obtained unexpected results that you could repeat? What were the consequences of that finding? Anyone here ever have that happen to them? All your experiments always worked out exactly right? I've certainly had this experience. I'm hearing, I think, a murmur of agreement. How many people have ever had an experience of getting something that was unexpected and it turned out not just to be an error, but to be really something that was reproducible? Not yet? Well, it will happen, trust me. Okay, if you were in Jocelyn's place, how would you respond to Professor Collins' inattention to her findings? Do you think that the professor here should be corrected? What do you think? Well, here. Anyone? Yeah. I mean, clearly, what's happened here is you can see that her experiment and the standard experiment worked just about exactly the same. That's probably just experimental error. But there's all this curve here, this inhibitory phase. Do you think that can just be ignored? Any other thoughts? Well, I think there's something to be said for what the professor recommended, in that it's true when you're doing a comparison of a bunch of compounds by some assay, you want to have that assay standardized. And this clearly is the range where it's standardized. But if you're really studying how those interact with the system from the point of view of mechanism, it's hard to ignore the rest of this curve. Something really remarkable is happening there. What would be an alternative to simply ignoring this curve? Okay, well, let's say she repeated it, she has repeated it twice, this report says. Let's say she repeats it and she gets exactly the same result. So what she's shown here is clearly that the standardized experiment works exactly the way it's supposed to, but if you go to much higher concentrations, you get something very different. And let's say this is now, they're ready to write up a paper about this material. Do you think this should just be left out of the paper entirely, or do you think something should be said there? I think so too, yes. Actually, even if she doesn't understand it, although it would be nice to try to understand this, and we'll talk in a minute about how this might have happened, this would be the sort of thing, what section of the paper might this actually go into? Well, it could go into supplemental data. I think I'd be inclined to put it somewhere else, though, in the published part. Which part of the published part of the paper might it go into? How many people think introduction? Materials and methods? Results? Discussion? Maybe discussion. That'd be a possibility. although you can't really put data in the discussion, so your data probably has to go somewhere else. I might even be tempted to put this in maybe at the very beginning of results, perhaps even as a table footnote or a figure footnote, or perhaps in the methods. To say, look, we're doing this assay under these conditions, but we have observed that when we use higher concentrations, we observe inhibition. The nice thing about doing that is, first of all, one of the ways that we rank papers very often is by how often they get cited. And I'll never forget when I was a graduate student, my advisor said to me, if you really want to have your paper cited the most, probably the best thing you could do is invent a new protein assay. And you will become a citation classic. So, you know, if you've discovered something new about this method that is widely used, and you publish it, a lot of people may notice that. A lot of people reading this who may not care particularly about this SR compounds might care a lot about this assay. And that might be very important to other scientists who might not know about this range. So I think having the method covered there is very important. So I think at a minimum, I would present this. I don't think it necessarily deserves its own paper, but it certainly may need to be presented. Now, if we assume that she proceeded with the experimental plan, which consists of testing a group of novel compounds for the stimulatory and inhibitory actions on nitric oxide synthase at 0 to 1 nanomolar concentration range, and a month later she completed the work and was asked to write it up. How should she write up the standard experiment given her doubts about the narrow range curve? Well, I think you've really already answered that. She'll have to just explain what this is. Now here, in the bottom of this page, you can see here what the answer probably is. In her continuing review of the literature, Jocelyn discovered that a very recent paper indicated that nitric oxide, the product of the reaction, inhibits NO synthase when present in high concentrations in contained systems such as the one with which she was working. When shown the paper, Professor Collins dismissed it and asked Jocelyn to write up the experimental results as she did them. What are Jocelyn's obligations in writing this up, and to herself and to her mentor and to science as a whole? Okay, so having found this paper, that puts her in an entirely different situation. Now she not only clearly has results, but now she has an explanation, right? That when you put in a lot of this compound, you may create additional nitric oxide that inhibits the enzyme, so you see this inhibition. So now she not only has a result, but she has a citation that can explain the result. This removes perhaps what would be the one reason that I might consider for not publishing the result. Because after all, one of the risks could be she could do very good work, let's say, in this low range of concentrations, and she might say, look, but I observe this strange thing over here, and I don't know why I observe it. Now, a journal editor might go back to her after she submits it and say, well, you've observed something very interesting. Why don't you find out why that happens, and when you find out why that happens, then resubmit the paper. So now they can't get their data over here published because they can't do that now until they do this. So that's a risk in submitting this data. On the other hand, if you can say, look, I observed this, and I found something in the literature that explains why this probably happens. Now that removes that objection, and in fact makes that paper very important. So here, I think I would, as a student, it would be very important for Jocelyn to argue with her advisor. How can a student argue with an advisor? Is that something that's possible? Well, with a good advisor, it should be. If you ever find, sometimes though, you know, students and advisors can have disagreements where just one feels one way and one the other way. So one of the things that I think is always a good idea is to have laboratory group meetings so the whole group can discuss these things. And probably every student should, early in their careers, have a research advisory committee that's helping them with their research. And that advisory committee, again, can be a forum where if the student and the advisor have a disagreement, they can bring bring that to the committee. So that's something you may want to think about, and we'll be discussing later on when we talk about collaborations. Okay, I think I'm going to skip this one case here, but we can go to it later. Let me just go to that. Well, here's one. This, I think, really relates to the ownership of research materials that I think is important for us to talk about. Ronald Wu had just completed the requirements for his PhD, and his thesis defense had gone extremely well. Dr. Morris, his mentor, wrote a letter of reference in which she said, "Ron is the best graduate student I ever had. He should be justly proud of what he has accomplished during the 5 years he spent in this lab, having nursed a very important problem in transcriptional regulation from conceptualization to the purification of several key transcription factors and the cloning of their genes. Furthermore, one of the materials he prepared has the potential of being an important therapeutic agent." Based on the basis of his performance and recommendations, Ron was accepted into a prestigious and comparatively well-paying postdoctoral fellowship in a productive, highly regarded laboratory in his area of interest. This situation was particularly attractive since it held the promise of a faculty position in 2 years. During his recruitment visit, the lab chief, Dr. John Link, suggested that the more materials Ron could bring from his current laboratory, the more productive Ron could be, avoiding the time and expense of preparing the materials anew. Up until this point, Ron and Dr. Morris had not discussed the disposition of the products of his research. One day, Ron met with Dr. Morris to thank her for all her years of support and friendship and for helping him obtain the fellowship. When Ron raised the topic of research materials, Dr. Morris restated her affection for him and her appreciation of his promise as a scientist. Dr. Morris then stated, somewhat apologetically, "Of course you understand that the materials associated with your research project belong to the lab. I really can't allow you to take your notebooks, probes, or other materials with you. I'm sorry, Ron, but that's just the way it is." So, what do you think of Dr. Link's idea of hiring a postdoc and having him bring all that great stuff from another lab into his lab? Okay. Someone from the back there. How about you? Last row? What do you think of that idea? Is that a good way to advance science? We'll simply find a lab we want to compete with, and we'll hire their graduates as our postdocs and say, empty out the freezer, bring it all to us, and we'll continue that work? Anyone? Is there a problem with that? I'm seeing some nods. Well, there obviously could be a problem here, right? I mean, Dr. Morris is not ending her research when Ron leaves. And so now that's one problem. As we're training people, we're training our future competitors. I hope we're training our future collaborators too, but we are training our future competitors. So, as we discussed, all that material that Ron created doesn't belong to Ron. But in actuality, it doesn't even belong to Dr. Morris. It belongs to the university, right? So, technically, the university might say, okay, this work might lead to patentable material. We don't want to let it out. We want to keep it. Does anyone see any problem here? Do you think this is fair to Ron? What do you think? How would you feel if you were in this situation? Well, again, one of the things I think this story suggests is it's very important early in the course of your education. This is something you might want to talk about your advisors after this weekend. Is you're all planning on leaving someday, and it's really worthwhile for you to think about who owns your research materials and what might be a fair way for you to continue on, what could you take with you, and what you can't. Now, I must admit I've never really been in this situation, but in most laboratories it's quite common to allow the students to take copies of their notebooks with them. But in fact, you're not really supposed to take the original notebook with you. You're supposed to leave that notebook in the lab, because if you've done research in the lab and that work has been published and a question comes up about that publication after you've left the lab chief is supposed to be able to access that data. And if that data is with you while you're doing a postdoc out in California, that lab chief can't answer that question. So a pretty standard way of handling this is to leave the original lab notebooks in the lab and to let the student take a copy. And when students have left my lab, I certainly have often allowed them to take with them research materials as well, although I have not allowed them to take all the materials and leave me with none, since I intended to continue on that research. But they could take a portion of those materials, of cell lines or of mutants or of DNA probes or things of that sort. That's certainly reasonable. But again, that might depend on the situation. If you were working in a lab, let's say, where it was a highly competitive situation, or where a patent application was planned but had not yet been submitted, and there was an issue of whether that should be released, then it's very important. It might be very important for that lab not to allow that material to be disseminated. And that could be an issue, so you have to keep that in mind. Go ahead. And that's a key thing. So let's say he's already defended his thesis. Well, the thesis is a publication. So everything that's in the papers that he's published and everything in the thesis has been published. How about that material? And I think when we think about that, we get to this paragraph here. And this could be an argument that Ron could have used. Ron could have said, there's a simple precedent in other areas to support the notion that I should have access to these materials. For example, Cell and Science explicitly state that materials must be made available to qualified investigators. In other words, if you write a paper, this is true for any journal, if I write a paper and I describe research I've done on a cell line, the assumption is made that I will provide that cell line to anyone who wants it. I may tell them you have to pay the postage, but I've got to provide that material. Since I did the work, I must be a qualified investigator. Also, NIH requires that materials developed under NIH grants be shared. Ethically, you cannot deny me the materials. Furthermore, as a graduate student, I'm entitled to share in the profits derived from my research as you are. Now, what do people think of this argument? Think that, but on the other hand, this argument does not say, again, I'm required, let's say, if I have a cell line, I have to share that cell line, but if I purify a protein and I've worked purifying that protein for a year and I now have a few milligrams or maybe micrograms of that protein in my freezer, and someone says, I'd like that protein in your freezer, I don't have to give that protein away. I could say, here's the method by which I purified it. You're welcome to purify it. But I spent the year purifying. I'm keeping it in my freezer. I'm not giving it to anyone. But if I purified a particular cell line and that person doesn't have the cell line, I have to give them the cell line. So Ron can't pull up with his pickup truck and empty the freezer from the lab. But yes, he would be entitled to take certain materials. And but again, some he would not be entitled to. But again, a very good idea to to have these discussions in advance before you leave a lab. The earlier you have the discussion, the better it can be, because different labs will have different circumstances, and it's important for you to realize what circumstance your lab is in. Now, as we discussed previously, you can and often do omit data, and not all data has to be published, and that is not necessarily falsification or fabrication. I'd say of the people who come to me, at the time that I was Associate Dean of our graduate school, I would often be the first stop for students who felt that they had somehow observed misconduct in their laboratories. And an interesting phenomenon that I've noticed over the years, I don't know if that's universally true, but it seemed to me that the less experienced students were much more likely to have questions about whether scientific misconduct had occurred than the more experienced students. Now, some might say, well, that's because the new students are really honest and they haven't been jaded by exposure to the establishment. But I don't think that's really true. I think what rather happened is that this whole question of how do you omit data is something that newer people in science have a hard time with. And it's something that you'll learn over the course of years. I'm not going to give you the ultimate answer on how to do it, where the more experienced students are more able to come to an agreement with their advisor. But again, if there are disagreements between researchers, the best thing is to discuss them early and discuss them openly. And I think most advisors are really very open to this. But as you'll see, you're never going to be in a situation where you can present all your data. Okay, here's a case that kind of exemplifies that. Alan Yeager has completed a series of experiments characterizing the receptor for a new class of hormones. During the course of his work, he studied binding characteristics and hormonal responses in tissue culture and in vitro, utilizing gels to characterize the molecular weights of receptor variants. This was exciting work for a second-year graduate student doing his first project. One day, Alan's laboratory chief asked him to prepare an abstract for an upcoming meeting and a paper for publication, both to be based on the work Alan had been doing. The abstract was due in 1 week. Often the source of ethical problems when there's a time constraint. As Alan examined his accumulated data, he noted that a number of cell plates failed to respond to the hormonal stimulus, and there was a considerable variability in the dose-response relationship. And here you can see the data. So here you can see it was a nice relationship, but clearly there are a few points, especially these points over here, that just don't seem to fit very well. I'm sure all of you have seen data like this. I know I have. Furthermore, on re-examination, he noted that a number of his gels were not very aesthetic in appearance, yet he was sure they demonstrated the molecular weight, agonist binding, and subunit characteristics of the receptor. Alan mentioned his distress to Pam Alden, a fifth-year graduate student, who said, "Why don't you clean up your data? You'll never get the paper published unless you do. We always clean up data around here." She then suggested that the 4 culture points failing to show a response along the x-axis be dropped because the cells were probably dead. Here, we'll go back to that. You can see, like, these guys over here. I could be convinced those are dead cells. She also pointed out that he might eliminate the top data point at the 45-minute interval as an outlier. She examined the gels and suggested retouching the negatives from which the prints were to be made, including the duplication of one of the nicer gel lanes to replace another that had turned out poorly, but showed essentially the same result. "This will greatly improve your chances for publication," she said. Alan replied, "Maybe I should repeat a few of the experiments or try to improve the culture conditions." "No," said Pam. "If you're convinced of your results, why go through the time, expense, and uncertainty of more repetitions? You'll never complete an experiment in time for the abstract anyhow." Somewhat dismayed, Alan thanked her and turned back to his work. What do you think about Pam's comments on publication practices and suggesting about cleaning up the data by retouching gels? Anyone think that's a good idea? Good, good. Because this may have once been true. However, nowadays, journal editors have been burned by this a number of times. And there are a number of computer programs by which they can look at these data and check to see if they've been touched up. And if they have been touched up, they ding you rather badly. So you don't want to do that. And in terms of those data points, how many people vote for just leaving these out and saying nothing? How many people vote for publishing the graph with these points in it? Does anyone think if you publish it with these in it, there should be some explanation of them, maybe in a figure caption? Yeah, I think so. I think the two acceptable things here would either be to publish these points and say in a figure caption, we believe that these were dead cells. Of course, the best thing would be if you would stain them and prove they were dead cells, but that probably wasn't done. Or I think a second thing that might be acceptable, but I think would probably be not quite as nice, would be to leave them out, but say, you've left out, one culture of which all the values were 0, which was you believe were dead cells. But there's certainly nothing wrong with having things like that, and there's also nothing wrong with having something like that. You can comment on it, but probably your best bet is to comment, and you could either leave it out or not leave it out, but you should never do that without adequate explanation for what you've done. And I think publishing it may be the best thing. So it's very important that we remember we can't clean up our data, except the best way to clean up our data is by repeating experiments. Now, it is permissible to take gels and cut them and show 3 lanes from one gel and 2 lanes from another gel. But when you do that, you can't simply do it with your computer. You do it, you say, panel A shows the results from a gel, panel B shows the result from another gel done under similar conditions. So the reader can see what you've done. It's perfectly all right, but what you can't do, and this was something that used to be done, is people would say, well, I'll take this lane from this gel and this lane from this gel, and I'll put them together and it'll look like it's one gel, and no one will know the difference. That's not allowed. So that's very important for you to realize.